Hello everyone, welcome back. For this week's experiment, we will be doing a PCR product purification and restriction enzyme digest. You can follow along with me in your lab manuals on page 18. So remember last week we ran a PCR and we obtained our PCR product for the beta-gal gene. So now we need to purify all of our beta-gal gene away from all of those free nucleotides and salts and other cofactors that were in that mixture. So that is why we're going to run this PCR purification. The kit that we'll be using to purify our DNA is this nucleospin gel and PCR cleanup. So to begin, I already have in this tube 80 microliters of binding buffer. This buffer will help our PCR product to bind to the silica gel in our spin column. And I'm just going to mix in 40 microliters of our PCR product. So this should give us a total volume of 120 microliters. I'm going to make sure to mix thoroughly. All right. So once we have our PCR product thoroughly mixed, I'm going to transfer the entire volume into a silica spin column. So right here, you can see is our silica spin column. It detaches into like a waste tube, and this is our actual column. So if you look closely, you can see that the white underneath that yellow ring is the actual silica. So that is where our DNA will bind to. So I'm just going to draw up the entire volume inside my tube and just transfer it into my silica column. All right. So the next thing I'm going to do is place it into the centrifuge. And make sure to counterbalance it with an appropriate volume. So remember it was about 120 microliters. So this is just a counterbalance. Sorry about that. Forgot that I needed a top. So I'm just going to place this top down, close the centrifuge, and we're going to spin it. Let's see. So we're going to spin it at max speed, so 13.4 RPM for 1 minute. So as the centrifuge spins, it'll draw all the liquid out of the silica column, and as it pulls the liquid down, the protein will get stuck inside of the column. All right. So now that our column is able to spin for that 1 minute, I'm going to remove our column. And you can see down here that all the liquid has transferred to the bottom of the collection tube. So the next thing I'm going to do is add in 200 microliters of wash buffer to the column. And we're going to make sure that we do this wash twice. So here is our wash buffer. Remember, this wash buffer contains ethanol to remove all the residual salts that might be stuck to the column. All right. Let me make sure the counterbalance is correct. Okay. and we'll do it for the same amount of time, at the same speed. So I'll just do this for another run, and I'll get back to you guys after we are done with the wash step. All right. So now I have completed washing our column. So you can see that we've washed it about 2 times. There's a ton of liquid collected at the bottom of the collection tube. So the last thing we need to do is just remove our column from the collection tube. So our DNA should still be in this column. And we're actually going to put it into an empty Eppendorf, like so. So now what we're going to add is 35 microliters of elution buffer. This elution buffer will allow for our DNA to solubilize and exit the silica column. So I want to make sure that when I add the elution buffer, I add it right at the center of the silica matrix. So I want to let this sit for about a minute. This way I can allow for the buffer to kind of help solubilize as much DNA as possible so we can just collect all of it. It'd be a shame if we just put it in and immediately spin it down and we only collect about 1/3 of the DNA we were supposed to. So just let that sit for a little bit. All right, so a minute has now passed, and I'm going to add it into our centrifuge. Make sure that our counterbalance or counterweight is the appropriate weight. All right. Make sure that they're as far apart from each other as possible. And just like all the other steps, I'm going to spin it at 60 seconds at 13.4 RPM. All right, so we've finished up here purifying our beta-gal DNA. So you can see in this tube is our 35 microliters. So now what we need to do is start our digestions. So we're going to be digesting both our pure beta-gal gene as well as this PET30A vector, which will be the plasmid that we attach our beta-gal gene into. So I'm going to be using the same 35 microliters, or I guess the entire 35 microliters of our beta-gal DNA. And into that, I'm going to be adding 8 microliters of water. All right. Then 5 microliters of 10x buffer that will help the digestion occur a little more properly. So here is our 10x buffer. And the last things I'm going to add are our restriction enzymes. So remember that these are enzymes, so they are proteins. We have to keep them on ice, or else the restriction enzymes might lose their function. So this will be 1 microliter of our first restriction enzyme, HIND3. as well as another 1 microliter of our other restriction enzyme, BAMH1. So with that all together, in that tube or in our digestion should be about 50 microliters. I'm just going to leave it in the ice. And the next thing I'm going to do is start setting up the digestion of our plasmid, or the PET30 vector. So for this one, I'm going to start by adding 35 microliters of water. Or 33 microliters of water, sorry, 33. Into this empty tube. And next I'm going to add 10 microliters of our actual vector, so the PET30 vector. So remember, because each microliter has about 1 microgram, that means I'm going to be using about 10 micrograms of PET30A. Mix that in. Next, I'm going to add that 5 microliters of the 10x buffer. And then again, the last 2 components will be our restriction enzymes. So starting with, I guess, BAMH1. And then HIND3. So with that, we now have both our digestions ready to go. We just need to make sure that we incubate them at 37 degrees for about 20 minutes. This will allow for the reaction to occur. And so once that is done, I will start setting up the gel for the gel extraction. All right, so it's been about 30 minutes since the incubation. Both our beta-gal as well as our PET30 digestion are now ready to go into this gel. So you can kind of see that there's already things inside the gel because I already added everything in. But something happened with the recording, so I'm going to kind of explain the process once more. So in the very first well, we have, just like last week, a molecular ladder, so we can compare our PCR product as well as our plasmid digestion to this ladder to determine if it's the right size. In the very first well, I have about 5 microliters of our pure beta-gal gene that we digested, mixed in with 5 microliters of an orange dye so that you can see it inside of the gel. And in the second lane, or I guess in the third lane, I have the digestion for our PET30, so 5 microliters of this, with another 5 microliters of the orange dye. So I added both of these into these 2 wells, and I'm just going to let them run for about 30 minutes at 120 volts, so very similar to last week. So from this gel, what we're mostly trying to see is that if our stuff was actually digested. And this will be useful for us in the future so that when we know that it's digested, we can actually combine these 2 and ligate them so that we can actually have a plasmid with our gene in the middle. So I'm just going to run this gel, and that'll be pretty much the end for this week's experiment.